Hey, so we are continuing on with the Unit 3 tissues, looking at epithelial tissues today. So what are the major functions of any epithelial tissue? It could be for protection. Generally, these are going to be cells that rapidly divide. So like your skin, your hair, your nails, your stomach lining. One of the things to think about with epithelial tissues is like when you think of the signs of chemotherapy, right? Person loses their hair, their skin gets bad, they get sick to their stomach, they vomit a lot because chemotherapy, or at least it used to, they've gotten a little bit better, attacks rapidly dividing cells, which would prevent, like that would go kill a tumor. But because it's finding rapidly dividing cells, it also hits some of the ones that are naturally doing that in our bodies. Okay, so anything that comes into contact with the environment is going to be lined with epithelium. So within your lungs, within your whole gut tube, so not just the stomach, like the rest of it as well. Like your little membranes of your eyes and stuff like that. Okay, other functions, absorption. So within your digestive tract, we'll have epithelial cells that absorb the nutrients into your body. Secretion in your glands and ducts. Hormonal glands are surrounded with epithelial tissue as well. And then excretion, wastes that actually leave the body versus secretion, where it does not leave the body. Okay, so excretion, sweat, oil, mammary glands. We're going to have goblet cells that release mucus, and those will be like lined throughout the digestive system. Okay, conduction of secretion. So within your blood vessels, getting the we'll get it. It'll have its own slide later. Filtration. So within your kidneys, diffusion. Remember, in your lungs is where the gas exchange between oxygen and carbon dioxide with the external environment and your blood vessels occurs. So that happens across epithelial cells. Transport, actually moving stuff around. So like there's epithelium in the ovaries that actually like do this come hither motion for the egg to get into the fallopian tube for fertilization or potential fertilization. And then hydration. Our epidermis produces oil to prevent evaporation of wet tissue below. So like we have like oily skin is protection so that we don't just like dry out every time we go outside. General features. They are usually 1 to 5 layers thick. If it gets to be more than 5 layers, then those top layers are usually dead. Everyone go like this. You see like little flakes come off of your skin. It's not like you got dandruff on your arm. That's normal. Okay, those top layers, everything you can touch of your skin is dead because it's more than 5 layers thick. It's usually somewhere around like 30. And diffusion of nutrients can only go so far. So all those dead ones on top stay there and form a protective layer, but nutrients can't penetrate to them, and so they die and they sit there. But that's kind of the nice thing about having that oil on your skin too, is that it helps attach them onto your skin, and they don't get away, and then you have that extra protection from the external environment. They form over a layer of connective tissue, so like the fat underneath. They line and cover body passages and organs. So all of your digestive organs are lined with this. They act as a boundary. They have one surface exposed to the passing materials. That's called the apical layer. So one side that stuff goes over it. Okay, so apical surface, what I was just saying there. This is the side that's going to be furthest from the basement membrane, whatever its attachment point is. And then lateral surfaces are going to be the ones that are nearest to the basement membrane. And they're going to face whatever other tissues are surrounding this area. They are non-vascular. So if you get a cut on your hand, but it doesn't bleed, right? Like a really like light cut. It's probably that you only went through the epithelial layers and not any lower. They allow for rapid healing, reproduction, and renewal. As I was saying back with the chemotherapy, that they are rapidly dividing cells. Generally, speaking of your skin specifically, and I'll talk a lot about your skin specifically here just because we can see that one. Generally, every 2 weeks is when you have new skin, new skin. That's about how long it is that they divide and make new ones. And then polarity. As we move up those layers, the cells look different than they do in the lower layers. So looking here, we have that apical surface. This is the top part. Stuff would pass over up here. The lateral surfaces are the ones between the adjacent cells. All of these here, these pink guys, are epithelial cells. We've got a basement membrane that it's attached to. There'll be nerve endings that go up through there. And then the connective tissue is where the blood vessel is, but notice that there's no blood vessels actually up in the epithelium. Ways that we classify epithelium. They can be simple, meaning one layer, stratified, multi-layered, or pseudo-stratified. Looks like it's got a lot of layers, but it actually doesn't. So that's how we can classify it by layers. We also classify it by shape. Squamous looks like scales, cuboidal, little cubes, columnar, tall columns, and transitional is just anything else. That's all they call it, transitional. It's not very common. Mostly we just find it in our bladder, so it's nice. Nice that we don't have to like really keep that together. Simple, pseudostratified, stratified. So one layer looks like a lot of layers because of where the nuclei are located, but actually all of them touch the basement membrane, and so it's single layered. Versus here, stratified. There are all these guys that touch the basement membrane, but then there's all these that also don't touch it, and so that's what makes it stratified. And then squamous, notice how they are like scale-like versus cuboidal, these nice little puffy pillows. And then columnar, tall and skinny ones. Again, stratified is just, it doesn't fit into those, not stratified, transitional doesn't fit into these three categories, and so that's why it gets its own name. So, depending on their function will depend how many layers they have. So for simple epithelium, generally these are going to be the ones that do diffusion, osmosis, secretion, or absorption. We only want the one layer so that we can quickly get to either gas exchange or nutrient exchange. Stratified epithelium is where you have 2 or more layers. And generally these are for like wear and tear. So like if you think of like kind of like the movement of things going across each other. And then pseudostratified, just because the nuclei are located at different levels, you can go back to the previous slide. It looks like it has a lot of layers, but it doesn't. All of the cells are attached at the basement membrane. They don't have to reach the apical surface, but they are all touching the basement membrane. And then for the different shapes. So for squamous, they're very thin, arranged like floor tiles. They have kind of a scaly appearance, allows for rapid passage of substances. Cuboidal, they are cube-shaped, right? Oidal just means like representing or resembling. May have microvilli on them, little tiny hairs. Okay, generally for secretion or absorption. Columnar, they look like tall columns. You saw the pictures of them. May have cilia or microvilli, those little finger-like parts. Generally for secretion and absorption. And then transitional, they could have any of the shapes and they can change shapes. Generally, when we think of them, it's in the urinary bladder for stretching and collapsing of the bladder. It's not like commonly found in other places of the body, so which is why it's only mentioned like the one time. Notice it's not, showing up in all these other pictures. So there's going to be a histology packet that we're going to label the different types of tissues. Okay, so we're going to look at simple epithelium, simple squamous epithelium, looking at our different endothelium versus mesothelium, and we're going to label all of these within that packet. So I will give you an empty packet, and then you can, let me go back here, and then you can go back through and label them yourselves if you want to print it, or if you want to do it digitally, if you've got like a way to mark up and like write on the papers. Okay, so just going through all of these, this is all going to be in that packet. We're going to be labeling. I'll slow down, sorry. Labeling the different parts of the packet. I will post an answer key to that as well, but do the actual labeling. Don't just look at the answer key. You need to actually go through and do that labeling, do the practice, so that you can be able to identify what shape the tissue is, how many layers it has, and based on that, be able to figure out its overall function. So that's what we'll do next. Okay, can't touch the pause button. There it goes. Oh my goodness. Hey.